The effect of Bacopa Monnieri extract and EDTA on seizure activity in N2 and CCA1C elegans. The research question that is being addressed in this investigation is what is the effect of different treatment methods, Bacopa Monnieri and EDTA, on seizure activity in CCA1C elegans, which is a mutant strain, and N2 wild-type C. elegans. A seizure is a physical behavior after abnormal electrical impulses occur in the brain. It basically leads to uncontrollable shaking and muscles contracting and relaxing. The calcium ion channel is an important area of research, specifically the T-type calcium ion channel, because it's involved with neurons, and it has been found that increased calcium activity through this channel leads to excitability of neurons, and this excitability state is what can lead to seizures. So CCBs, or calcium channel blockers, could possibly be used to treat epileptic episodes by regulating the flow of calcium ions through this channel. A study that I modeled my experiment on tested Bacoside A, which is a chemical found in the Bacopa Monnieri plant, on N2 and CCA1C elegans. So they induced a seizure, and its activity was observed, and they calculated a seizure index, which is basically a measure of how intense the seizures were. And what they did was they tested the number of convulsions, which is basically an abnormal movement pattern in worms that basically indicates that a seizure is taking place. So the test showed that the Bacoside A compound reduced seizure activity. They also found that the CCA1 worms had no convulsions. In my experiment, I used Bacopa Monnieri extract because it contained the Bacoside A compound. So this was the graph for their experiment. They found that at the highest concentration of Bacoside A had the least seizure index, which means that they basically worked the best to reduce the intensity of seizures. The CCA1 worms, as I said, did not experience any seizures, while the wild-type worms did. And this indicates the critical role of the T-type calcium ion channels. The CCA1 worms lack the CCA1 gene, and this gene basically produces the CCA1 protein. And this protein makes it easier for calcium ions to flow through this channel. So since the CCA1 mutant worms do not have this gene, it makes it harder for calcium ions to flow through the channel, and which is why an excitability state was not triggered, because the calcium ions were basically blocked. EDTA, which is another chemical, which is the other treatment that I decided to use, is basically an ion blocker that inhibits the flow of ions differently to Bacoside A and reduces the effects of ions on the body. I use C. elegans in my experiment because of its advantages, such as it's small, easy to grow and maintain, it requires little food, they reproduce quickly, and ion channels and mechanisms are similar to humans. For example, the CCA1 gene is actually an ortholog of a human gene, which means that basically, based on the responses of the CCA1 worms, can be indicative of how humans will respond to treatment as well. My hypothesis is that the Bacopa Monnieri extract and the CCA1 mutant C. elegans will produce the least amount of seizure activity. This is because both have been shown to reduce seizure activity by regulating the calcium ion channel effectively and directly. The EDTA treatment, on the other hand, does not specifically target the calcium ion channel. And so CCA1 worms also have a genetic advantage by making it harder for an excitability state. The main objective here is to learn about potential treatments for seizures in humans, because there's no treatment for epilepsy and it's quite common. I have two independent variables here, which is the treatment method and the worm strain. The dependent variable is the seizure intensity, and this will be judged based on a quantitative measurement, which is a number of unilateral body bends, or convulsion, which, as I said, is just an abnormal behavior pattern. And qualitative is the seizure intensity, which basically includes things such as speed and overall intensity. I had a negative control, which was no treatment use, and I have my constants here, which are things that I try to keep the same to lessen their effects on the on my results, which I assumed in my analysis of the results that these things were kept constant. I had 3 trials of 10 worms for all 6 combinations of treatment method and worm type. The method of measurement, as I said, was the number of unilateral body bends, which is counted under a microscope. For my statistical analysis, I used t-test, standard deviation, and mean. These were the materials that I used. The thermometer gun was unique to my experiment, and I'll explain how I used it, my procedure here. So I made 24 plates of NGM agar and OP50 broth. OP50 broth basically provides food for the C. elegans, and agar is a medium for them to grow on. Of these 24 plates, I split up into 3 groups for each treatment method. So for 8 plates, I coated them with Bacopa Monnieri extract. The other 8 plates I coated EDTA on them, and then another 8 plates I kept blank for the no treatment method. After worms were chunked onto these plates, I let them grow for 48 hours, and then for the actual experimenting part, I put these plates on a hot plate and gradually increase the temperature until about 30 degrees Celsius. And this is where I use the temperature gun to basically allow me to monitor the temperature of the hot plate and of the petri dish. So when it got to 30 degrees Celsius, I would put 1 drop of a buffer solution, and this ionic solution basically increases the amount of ions in the worms and induces a seizure by causing an overload of ions. Then I immediately transferred the plate to a microscope, where I observed a single worm at a time, and I would set a timer for 10 seconds and record the number of convulsions that I saw. And then I would record this data and return the petri dish back to the hot plate and repeat this for 10 worms. So here's the data that I have. It shows the mean number of convulsions per 10 seconds for each treatment method for both types of worms. It also shows the standard deviation, the p-values, which were from the t-test that I used, and the asterisks basically show whether the results were significantly different or not. I also displayed a graph to show this, which basically makes it easy for you to see the clear trend here. I have a blue bar to represent the N2 worms and an orange bar to represent the mutant worms, and I have the number of convulsions on the y-axis, and I also indicated significant difference with the asterisk again. So here you can see a clear trend that the number of convulsions increases from left to right. So Bacopa had the least number of convulsions for both types of worms. EDTA was in the middle, and then the untreated worms, as expected, had the most number of convulsions. Yeah, you can also see that the orange bars are consistently lower than the N2 bars for each treatment group, which basically shows, as predicted, that they have some genetic advantage that allows them to have less intense seizures. So the Bacopa treated worms had the least number of mean convulsions, with 3.43 for the N2 worms and 2.40 for the CCA1 worms. The EDTA bars were in the middle, and then the untreated worms had the highest bars. The same trend was observed no matter what worm type was used, and it's notable that the CCA1 worms always had lower convulsions than the N2 worms. The standard deviations also increased with the number of convulsions, which shows that as the number of convulsions per 10 seconds increased, the inconsistency and variability of the data did as well. The asterisks indicate a significant difference, and since I had a significant difference for every t-test that I did, it ensures the validity of my results and allows me to draw conclusions from it. So in conclusion, my hypothesis was supported. The Bacopa treatment recorded the least convulsions because it was able to inhibit the flow of T-type of calcium ions through the T-type calcium channel. Untreated worms had no method of regulation, which is why they had the highest number of seizures. The CCA1 worms recorded less seizures than the N2 worms, likely because they lack the CCA1 protein, as I said, which makes it harder to trigger an electrical impulse in the brain. And in a similar study, the CCA1 worms did not experience any seizures at all, and only adult worms were used in that experiment. And so for error, in my experiment, I did not control the age of the worms. So what should have been done was that only adult worms should have been used. but instead I used some younger worms and a mix of young and adult worms in my experiment. And as I realized later, this is not the best way, the most controlled way of understanding the true effects of these treatment methods on worms, because younger worms respond differently to older worms because they're not as mature and they're not as developed and they're not as able to defend themselves against a seizure. Which explains why in my experiment the CCA1 worms had some seizure activity, whereas in the experiment I modeled it after they did not, because they only use adult worms, whereas mine there was a variability of ages. So for further research, I would like to incorporate the variable of speed into seizure intensity, because what I observed was that some worms experienced seizures in which they moved at a more rapid pace of motion than others, but this cannot be accounted for in my experiment because it was not a given method of data collection. But overall, I felt my results were useful, and they indicate that the calcium ion channel is critical to controlling seizures, because when this channel was controlled, seizure intensity was less. And this indicates that perhaps something to regulate this channel would be an effective treatment in humans as well. Thank you.